So thank you very much for inviting me. I mean, as you can tell from a lot of the conversations we've had over the last couple of days, AI is very much in the middle of the conversation. What I'm going to hope to do is talk a little bit about kind of some of the things that are really going on. Not hypothetical, like someday AI will do some cool stuff, but what is happening right now. I'm Mark, as I said. Liz introduced nicely. I have a PhD in molecular biology. My undergraduate studies, though, were in computer science, and I did artificial intelligence research. That artificial intelligence research was in the 80s. It's early enough that both of my girls, who are professionals in artificial intelligence, tell me it doesn't count. And they're wrong. It totally counts. And my kids' reason for that is they said, well, Dad, nobody had computers then. I said, well, you're right. We didn't all have our own computers. All 3,000 of us shared a computer in the basement. Right? I think that wasn't helping my story. So I want you all to imagine together with me, like, what is a world like where some of the most difficult to treat, right now incurable diseases are becoming tractable? How is that happening as the result of people and machines working together in order to do it? And that doing so doesn't just benefit patients or the communities they're in or their caregivers, but as we heard from the previous panels, it's creating unique new opportunities everywhere. It is a democratizing force, and that is no more visible than in the healthcare and life sciences space. So we're going to explore this for the next few minutes. If you ask the question, well, geez, Mark, this new AI stuff has been 2.5 years? Where are all these new drugs I was promised, right? You have to remember this is embedded in an incredibly slow process, right? The development of new drugs takes on the order, on average, of about 10.5 years from discovery to treatment, right, of patients, 10.5 years. The record holder was 7.5 years. Sometimes, in the case of human gene therapy, that was the topic of my PhD work, it took almost 40 years, right? This is a long process. That said, shaving just a few years off of that process means innumerable people who are suffering from that disease and may in fact die of it have an opportunity that they didn't have before. This is a worthwhile endeavor, and it's one that even working at the margins can make a real difference. So I'm going to talk about a couple of examples, and they're at both ends of this spectrum, right? One end of the spectrum is making unique new tools that enable research and life sciences to go faster or more efficiently or discover new things. An example of this is our own Montana's own FireBio. They are a company in our portfolio. Two Bear Capital, as Liz mentioned, is a Montana company. So is Fire. At the opposite end of that space, I'll talk about Synavia Bio. They're opposite in the sense that they're actually developing the drugs and leveraging artificial intelligence to do so. Synavia is in New York City. So this is happening all over the place. So one thing about cancer in particular, but this is true for a range of diseases, Alzheimer's disease, Parkinson's disease, for reasons that we'll discuss, it's improving. It's getting better, but it's getting better slowly. In the early 80s, my mother had breast cancer, diagnosed with breast cancer, at a time when a breast cancer diagnosis was dire, right? It was bad news. That's improved substantially, breast cancer and prostate cancer. I will say, my mother is now 85 years old and riding her bicycle around everywhere, healthy as anything, right? I want everybody to have that opportunity to suffer from a disease like cancer and then 40 years later be riding around on your bicycle, right? That's what I want. The problem is some of the diseases haven't seen a similar level of improvement. Big diseases that affect a lot of people, colon cancer, lung cancer, Parkinson's, Alzheimer's disease, and even disease like pancreatic cancer that, although isn't as common as some of these diseases, has had almost no improvement, right? So the question is, how do we get these down here, up there, so everybody gets a chance to ride their bike? Well, the answer here is an interesting one, right? We're at the confluence of two accelerating paths, right? Technologies in general follow what we sort of call an exponential growth or a super exponential growth. In other words, we see this in everything, any technology. This isn't just computers, which we're all familiar with. Moore's law. Wow, computers get twice as good every 18 months. That's pretty amazing. Super exponential curve. These are log graphs, by the way. A straight line on a log graph is an exponential curve. Super exponential, where you see a growth like this, means it's steep. And we've seen this, right? Artificial intelligence is the best example. Why are these super exponential? You use the previous generation of the technology to improve the next generation of the thing. This is how computer chips got good fast. AI is getting good, but it's getting good exquisitely fast because we're not talking about a year between generations. We're talking about like weeks. That's why that vertical is almost straight up. This is why all of us experience this, wait, what? AI did that last week? Like it's new every week, something new. But this is true for fossil fuels, it's true for agriculture, it's true for anything, right? It's just the time span is getting compressed. The same thing, by the way, is going on in life sciences, right? This is a picture of the consequence of exponential improvement of technology, in this case, DNA sequencing, right? DNA sequencing cost goes down because the technology gets better. And how much it goes down? When I was in grad school, you got a PhD for sequencing one gene. There was a guy in the lab got his PhD for, it was like 1,000 base pairs. By the time I was a postdoc, they were working on the Human Genome Project. They sequenced the complete genome of 2 people at a cost of about $3 billion each, right? Now, the cost to run a full genome sequence is a couple $1,000s. That is about the cost of like an MRI series. Not surprisingly, it's becoming much more common to leverage DNA sequencing in diagnosis of diseases, right? And as a good example of this, like you go to any cancer center in the US, big designated cancer center is going to run your sequence for your tumor and use it as part of your diagnosis. This is common. But this isn't just true for DNA, by the way. You hear people talk about proteomics, they're doing that for proteins. Metabolomics, they're doing it for metabolites, and so on and so on. There's a massive increase because of the decrease in cost in the data available. What does this mean, right? Those paths intersect to create unique new opportunities. Compute at scale. We've got access to nearly infinite compute, thanks to our CoreWeave friends we talked earlier, nearly infinite memory and storage, and by the way, the near totality of human knowledge, anything that's on the internet, to compute on. What does this mean? This means that we can create new platforms to discover new things, like DNA sequencing at scale, for example. But then we can use that to do unique new drugs, targets and interventions, things to go after. The thing that's less well known is that that often then turns back around and improves those tools. And this is a closed thing. That closed loop we just talked about that accelerates technology, this is going on, right? And the yield here is faster and better therapies, but I remind you, it's embedded in this very slow process, right? And that's because of regulatory review and a bunch of other things. Those will improve too, by the way, but not as quickly, right? So let's just talk about 2 quickly examples. Let's talk about Fire Bio here in Montana. They've got one of these technologies that's unique and new. They're able to fish out what are called extracellular vesicles from the bloodstream. An extracellular vesicle is like a little envelope full of liquid from inside a cell that blebs out. And like an envelope, it carries a unique address, right? And that's because of the proteins on the cell surface bleb off with that. And now it floats around in the blood. This is a natural process, by the way. This occurs all the time. It also occurs as part of certain disease processes, especially glioblastoma, Parkinson's disease, Alzheimer's disease, right? But what that means is there are samples from these cells that, if you can properly find them, are floating around in the blood. You don't want to be drilling holes in people's head all the time to check how they're doing, right? It's bad for a lot of obvious reasons, right? But if you can just pull a blood sample and find out what those cells are doing, that's an accelerator for drug discovery and development. That means you can watch in near real time what your drugs are doing and how it's working. Well, what's really important about this is if you just look in the blood for things like protein and DNA, you can find it, but it's really noisy, right? The blood's a very noisy environment. And if you want to apply tools like artificial intelligence to that, you're going to have trouble doing it because artificial intelligence is a little like a radio amplifier, right? Like, we've all had this experience. You're listening to a faint radio station, there's all this static, and you turn it up. What do you get mostly? More static. Turning up a radio station embedded in a lot of noise doesn't really help, right? AI is that way. But if you can find a drop in the noise floor, because you're pulling very specific samples out of that, you can then apply artificial intelligence techniques. And just like turning up a loud radio station that's above the noise, you get more music. Makes it better. Same thing's true with AI, and this has been incredibly useful for Fire. Fire's working on right now on partnerships with a variety of pharmaceutical and biotech companies to help accelerate programs in glioblastoma, Parkinson's disease, Alzheimer's disease, and other areas. It's extremely exciting, right? The other end of this, of course, is the development of drugs, right? Well, okay, we're developing new drugs. Synavia develops drugs based on antibodies. Now, the interesting thing about antibodies, and this is common, lots of people develop drugs this way, is when you develop antibody drugs, you tend to generate hundreds of thousands to millions of them at a time. One experiment will generate a lot of antibodies. The problem is you can't go analyze 1 million antibodies. There's just not a way to do it. You typically do it at about 100 at a time in so-called high-throughput methods. As you can imagine, the way pharmaceutical companies traditionally do this is they go, well, all right, We'll just do it 100 at a time until we find one that works, and then into development, because you can't look at all of them. You can't explore the whole space, right? What happens is that you get drugs that work, but they work in 10 or 15% of the population. We've all seen this. Oh, I had cancer, and this drug only works in 15% of the people who get it, right? So the question is, how do you improve that? Well, the answer is you look at all of those antibodies, all million. Well, you can't do that in a lab. How do you do it? You use tools like AlphaFold, for example, Nobel Prize-winning tool used to predict three-dimensional protein structure. Or you use tools that Sanofi developed themselves to cluster, analyze, and identify what are the best antibodies from the whole set, every single one. And what that means is they can generate drugs that work better, more efficacious, but also can target many, many more patients. And we've seen this, right? And they're working with pharmaceutical companies right now who acknowledge, by the way, that the drugs that these guys are generating are superior to theirs. Right? What has this done? This has taken 2 full years out of the development process for those drugs. They're preparing to enter clinical trials with one of their drugs right now, 2 years quicker than they would have otherwise. More importantly, you know how many people are at Sanofi? Like 15. They're not a 100,000-person pharmaceutical company. This is what they were talking about earlier when they were saying artificial intelligence is a massive leverage point that lets a small group of very dedicated individuals who really want to improve the lives of patients do so. So, look, the thing I want you to walk away with is these aren't isolated examples. I don't want to say, 2Bear Capital is really good at figuring this out. We've got the only 2 companies in the world that are doing it. No, including those big pharmaceutical companies. Lots and lots of companies are doing this right now, and this means that there's going to be more and more impact in more and more lives across the board in this space. I don't want to light pedal it, right? And this was addressed earlier, but I want to make sure everybody walks away. Artificial intelligence is really disruptive, and we're going to have to manage that disruption, right? This is going to be a challenging thing like it always is. New technologies are always challenging. I do think that sort of Jevons paradox suggests that we're likely to see an overall improvement in jobs over time. But I don't want to light pedal the transition. But I do want people to walk away from that the gains that we're potentially getting out of this are not imaginary. These are real things that are happening right now, but the problem is they're masked because of the difficulty of the development process. So watch this space. Thank you for letting me speak. I appreciate it.